For decades, the study of memory has been fundamentally neuron centric. The prevailing wisdom held that the physical trace of a memory, the engram, was stored exclusively within ensembles of neurons. Today, we're analyzing a landmark study from Williamson and colleagues that directly challenges this foundation, presenting compelling evidence that astrocytes are not merely supportive cells, but are an active and essential component of the memory engram. We'll proceed through this analysis logically. First, we'll establish the classical neuronal engram paradigm. Then we will systematically examine the evidence for the role of astrocytes, following the paper's progression from initial observation to tests of necessity and sufficiency through to the underlying molecular mechanisms. So let's begin with the established framework, the neuronal engram paradigm. This has been the dominant model for memory storage for the better part of a century. The central concept here is the engram. It's not an abstraction, but a physical biochemical change in the brain. The hypothesis states that a specific ensemble of neurons becomes active during the initial learning event or encoding, and the reactivation of this very same ensemble is what constitutes memory retrieval. A key challenge has always been how to identify these specific engram neurons. The primary tool for this is the detection of immediate early genes, or IEGs. The gene CFOS, in particular, acts as a rapid molecular marker, essentially tagging neurons that have been recently active. This provides a crucial entry point to label and, importantly, manipulate these engram cells. The use of CFOS as a marker enabled the development of genetic tools like the CFOS-T-TA system shown here. This technology allows for the specific and permanent labeling of just those neurons that were active during a defined learning window. Crucially, these tagged neurons can then be artificially controlled, for instance with lighter chemicals, to directly test their causal role in memory. It's this approach that proved that reactivating these neuronal ensembles is sufficient to induce recall. This neuron-centric view, however, has largely ignored the potential contributions of other cell types. We know that astrocytes regulate synaptic transmission and exhibit their own forms of plasticity. This leads to the critical question Williamson and colleagues posed. Do astrocytes also form their own functionally relevant ensembles during learning? This brings us to the first set of experiments in the paper. The initial step was to establish whether astrocytes are, in fact, activated during a learning task, and if that activation has any functional consequence. Here in panel B, we see images from the hippocampus on the right in a mouse that has undergone fear conditioning. There's a clear increase in the red tomato labeled cells. These are CFOS activated astrocytes. Compare that to the home cage control animal on the left, where these cells are far more sparse. This is the first piece of evidence that suggests learning induces CFOS expression in a distinct population of astrocytes. Okay, so there's a correlation. But is it causal? To test this, the researchers informed a conditional knockout experiment, selectively deleting the CFOS gene only from astrocytes. The results are in figure 1H. Control animals on the left show high freezing, indicating strong memory recall, but the mice with CFOS deleted from their astrocytes show a significant impairment in recall. This deficit was also observed in other memory tasks, like novel place recognition, and was linked to deficits in synaptic plasticity. The conclusion from this initial part of the study is, therefore, unambiguous. Astrocytic CFOS expression is not simply a byproduct of learning. It is functionally required for both normal synaptic plasticity and for memory formation. So if these astrocytes are necessary, what exactly are they? The next section of the study focuses on characterizing this population, which they term learning-associated astrocytes, or LAAs, and their relationship to the neuronal engram. To study this specific cell population, a more advanced genetic strategy was needed. As outlined in panel A, they used a dual recombinant system. This allows for the permanent labeling of only those astrocytes that express CFOS during a very precise time window. This is the tool that creates the tag for the LAA ensemble, and these LAA are not just labeled, they are functionally distinct. Looking here at panels E and F, which show two photon calcium imaging, we see that LAAs have unique calcium dynamics. The bottom trace from an LAA shows signals with a significantly higher amplitude, a larger area under the curve, and longer duration compared to astrocytes from control animals. This suggests they are in a persistently potentiated state following learning. The next question was about the physical relationship between this LAA ensemble and the neuronal engram. Using a dual labeling strategy, the team tagged both LAA and engram neurons from the same learning event. The results were remarkable. First, the processes of engram neurons were preferentially located within the physical territories of the LAAs. Second, when they used a technique called eGrasp to visualize the synapses between engram neurons, they found these synapses were significantly more dense inside the LAA territories. This indicates a clear physical and synaptic affiliation, and the spatial link is mirrored by a functional one. Artificially activating the LAAs selectively increased the frequency of excitatory postsynaptic currents in their associated engram neurons. And in vivo activating LAAs boosted the reactivation of the neuronal engram itself. So LAAs aren't just near engram neurons, they're actively modulating their activity. This all leads to the central, most stringent test of the study. If LAAs are a true component of the memory trace, then their activation alone should be sufficient to trigger memory recall. The experimental design here is absolutely critical. It's not enough to show that activating some astrocytes triggers a behavior. You have to prove it's the specific ensemble. So they compare two conditions on the left, chemogenetic, activation of only the sparse LAA ensemble tag during fear conditioning, and on the right as a control, a bulk activation of a random widespread population of astrocytes. And the result was definitive. As you can see from the data in figure 4b, when the LAA ensemble was reactivated while the mouse was in a neutral context, the animal displayed robust freezing behavior, indicating memory recall. This effect was durable, lasting for at least 3 weeks. They had artificially induced the memory by activating only this specific astrocytic ensemble. The control experiment confirmed the specificity of this finding. Activating a broad random population of astrocytes as shown in figure 4f induced no significant freezing behavior compared to control. This is the crucial evidence that the memory information is stored in the specific pattern of the LAA ensemble, not in astrocytes of general. So we have established necessity and sufficiency. The final piece of the puzzle is the molecular mechanism. But what marks the identity of an LAA? What is the transcriptional signature that enables its unique function in memory? The researchers asked this to find this molecular driver. First, they used FACS to physically isolate the tagged LAAs. Then they performed RNA sequencing to identify differentially expressed genes. Using motif analysis on the genes that were upregulated, they searched for enriched transcription factor binding sites. One hit stood out significantly: the motif for nuclear factor NFIA. Having identified this factor as a prime candidate, they tested its necessity. They created mice where NFIA was conditionally knocked out, but only within the LAA ensemble, as the data in figure 5H shows. This specific deletion severely impaired recall of the fear memory. This identifies NFIA as a key molecular player required for LAA function. But this raises another question. Does NFIA deletion erase the memory, or does it just prevent the LAAs from communicating with the neurons? To find out, they performed a rescue experiment in mice with NFIA deficient LAAs. They bypassed the astrocytes and directly activated to neuronal engram. As you can see, this direct neuronal activation completely restored memory recall. This is a critical finding. It demonstrates that LAAs exert their influence through the neuronal engram, not via a parallel pathway. So, taking all of this evidence together, we can now formulate a new expanded conclusion about the cellular basis of memory. Let's summarize the key findings. Astrocytes form durable learning specific ensembles. These LAAs are intimately integrated both spatially and functionally with neuronal engrams. They are necessary for memory consolidation, a process that requires the transcription factor NFIA. Critically, the LAA ensemble is sufficient on its own to trigger memory recall, And finally, they function by modulating the activity of the neuronal engram. Fundamentally, what this work does is expand the engram concept to a neuron astrocyte ensemble. The line of investigation here is important. It precisely mirrors the progression of neuronal engram research over the last 2 decades, moving methodically from correlation to demonstrating necessity, and finally to demonstrating sufficiency. It applies that same standard of proof to astrocytes for the first time. This finding fundamentally changes our view of memory storage and opens up a host of new questions. If the boundary of the engram now officially includes astrocytes, it forces us to ask what other non neuronal cells might play an active role in storing information. Understanding how this newly revealed integrated cellular network contributes to the full complexity and robustness of memory is the next frontier. I want to thank the authors for taking the time to share their work publicly, and I really appreciate the opportunity to cover this fantastic paper on my channel.